Welcome back to Dr. Ghosh's Meet channel. Today we are going to discuss on space microbiology or impact of microbes for International Space Station establishment. The concept has been developed by Dr. Dipankar Ghosh as a part of student mentorship activity. First of all, I would like to acknowledge all the Microbes and also the participant of this student mentorship activity 2023, especially Diasha and Mohabrita, who have involved a lot to make this thing happen to all of us. The study of microorganisms in outer space is what we know as astromicrobiology or space microbiology. The existence of microorganisms in such an extreme environment, I would say in a microenvironment, has given a rise to the term extremophiles. The extremophiles, which means the organism that can tolerate extreme temperature, pH, salinity, and radiation. This has risen a big interest among the scientists and researchers in studying the interdisciplinary and interplanetary existence of microbes, usually occurred by the International Space Station. The large population of airborne bacteria, fungi were present, causing contamination in the International Space Station. The most dominant bacterial genus, so-called Staphylococcus, and fungal regime, so-called Aspergillus and Penicillium, they have been dominating this work done and contamination issues gradually being raised in International Space Station. Usually, these extremophilic microbes may even survive interplanetary transmission. Terrestrial Mars analog sites are the focus of researchers for understanding and comprehending the microbial diversity and detection of life on Mars. The bigger question here is, how do these microbes persist in space? It was analyzed that the serious damage caused by cosmic particles by breaking down the double-stranded DNA, which is being repaired by homologous recombination of the sister strands and non-homologous end joining method. So this is a nice pictorial presentation which have been showing how the double-stranded DNA breakage are the most serious issue and which is being damaged caused by AZE particle of the cosmos. However, the microorganism contain several molecular mechanisms to repair this phenomenon, where the strand joining by homologous recombination. Only a few microorganisms can fully cope with the space microenvironment. Multiple microscopic analysis has revealed that no detectable ultrastructure alterations were found in algae or the fungal cells, likely lichen or halophilic cyanobacteria. In the lichen symbiotic association, thick and dense cortex with the UV screening pigments, rhizocorpus, and the phenolic acid component present on the outer layer as a protective mechanism, whereas the salt crystals are present in an extragenous layer in case of halophilic cyanobacteria. Space environment can have a huge influence and impact on microbial physiology, metabolism, microbial drug resistance that is either increased, elevated in space, or decreased. Like in Salmonella and E. coli, the ability to survive in macrophage increases with the stimulated space conditions, while resistance to oxidative stress decreases, while in S. enterica, seroper, typhimurium, virulence and pathogenicity has a probability to escalate. So usually, there is a huge influence on the microbial morphology. The bacterial resistance is either increased in the space or overall drug resistance is decreased. As I mentioned, that is the oxidative stress. So you can see that how the microbial entity in the space environment, they are getting involved or getting exposure to multiple forms of extremophilic conditions like the thermal extremes, the microgravity, radiation, vacuum, that creates a huge stress response in those microbes. And that causes the membrane-associated read adjustments. And that influence the metabolic rearrangement that causes the alteration in the genome, and ultimately whether they will survive or they will dead. So this stress-responsive metabolic pathway exhibit reports that it's not found in natural conditions. Like virulence and pathogenicity, where evident has been shown that the probability of elevated pathogenicity and virulence that happen, as I told, in the Salmonella cerevisiae typhimurium. Under stimulated space culture, they're found to be more acidic resistance, suggesting enhanced survival possibility in the gastrointestinal tract. So this elevation in virulence is sufficient to alter the related pathogenesis. Although there is no definitive correlation till now has been established which regards how the space flight induces these altered variations in genetic expression findings. So microbes usually carried out by human and contaminant present in the air, in the water, and surface of the vehicle leads to contamination. So these contaminants may have adverse impacts on the avionics and system to the spacecraft. The biofilm formation in space is a common phenomenon caused by the surface microorganisms. Although with that, the crew are at a high risk of infectious diseases during the longer flight mission. On the brighter side, the microbes like cyanobacteria, fungi, they do contribute in performing various processes that include the vitamin production, water recycling, air decontamination, and the waste management. So this explains the risk of infectious diseases during long space flight missions. However, the immune response mechanism is not been fully studied yet. So biological-based life support system is for the long space habitations. A final note had been made for the beneficial uses of microorganisms and other biological systems. So biogenerative processes which mimic the Earth's natural biosphere offer increased potential for mass and cost saving compared to the consumable resupply. Plant provides suitable environment and edible biomass for the growth of bacteria, fungi, cyanobacteria, which are the candidate for performing various processes that include, as I told, the vitamin production, water recycling, and waste management system. However, this can be controlled by prevention of humidity, air filtration, routine housekeeping, and monitoring of the surface, water, and food. Various simulation and projects like Project Mika has been designed to ensure the health and the well-being of the crew during such kind of mission, like Mars mission. So there is this very interesting panspermia theory has been established to explain the interplanetary microbial transmission. So panspermia is the idea that organism can be transferred from one planet to another, originally discussed by Kelvin and Arrhenius. Rocks can be ejected from the planetary surface by colliding with the asteroids or the comets, harboring microorganisms. They do also carry out deliberation them in a variable state to other planetary bodies. This evidence of material ejection from the planetary space sets a firm basis for interplanetary panspermia. Pathway for interstellar panspermia are still unknown. The detection of shocked crystalline quartz can create for 104 kilometers away in evidence. Only a few notable microorganisms can fully adjust and cope with the space environment. Among these, Deinococcus redidurans, nicknamed Conan and the bacteria, was the only species to survive on the Martian surface. See, this microorganism is dried, get frozen, buried deep beneath the Martian surface. So these bacteria can survive in a harsh, non-permissive condition, such as dehydration, acid and cold temperature. Scientists have already been studied and have determined that this microbe is one of the most radiation-resistant organisms known to science. New studies determine that when Conan bacterium is dried and frozen, so they can bury deep in the beneath the Martian surface, it could survive for 140,000 units of radiation. These levels of radiation exposure could kill a human. Canon, the bacterium genomic structure, linked the chromosome plasmid together. The researchers were able to measure how many manganese antioxidant accumulated in the cell of this kind of microorganism as they were exposed to radiation. The more manganese antioxidants the team found, the most likely the microbe was able to resist the radiation and survive. Scientific research concludes that the terrestrial contamination on the Martian surface was permanent. As scientists say, this would complicate scientific efforts to look for the Martian life. Likewise, if microbes evolved on Mars, they could be capable of surviving until the present day. That means returning Mars sample could contaminate Earth. So these are the few references that will allow you to go for further study the topic of the microbial impacts on the International Space Station. Thank you very much.